Just going to talk about transcriptomic and atlas of the morphologic development of the fungal pathogen cocci. What do you know? Reveals key phase-enriched transcripts.
Thank you. And for all of the ecologists in the audience, I'm sorry, I am not an ecologist. I'm going to be talking about a lab strain. So I'm a clinical fellow in the SIL lab at UC San Francisco, and I'm going to tell you about this large-scale transcriptomic project we did that revealed some interesting biology. So I'm going to spend a moment to go through the life cycle since we haven't talked about it too much yet at this conference. But we all know Coccidioides is a thermally dimorphic fungal pathogen. That means it grows in the soil as hyphae, which produce asexual spores called arthrochonidia. And those, as we've talked about, are the infectious form that are inhaled. And once in the lung, they round and swell and grow into spherules, which develop internal septations, rupture, and those endospores that have now been formed inside are released, and each endospore goes on to form another spherule. And this beautiful life cycle has been worked out, but I think most people in this audience know that the real molecular underpinnings of the biology of most stages of this life cycle have been really understudied and are still poorly understood. And as you might imagine, that lack of knowledge of these different forms of the pathogen has translated into a lack of treatments that we struggle with our patients. And so I wanted to just ask the question, like, what transcripts are associated with each morphology? And luckily, I wasn't starting from scratch. Many people in this audience have done transcriptomic studies of Coccidioides already. Generally 1 to 2 time points of these developmental cycles. I think the majority of the studies have used different media to generate really robust hyphae and spherules for comparison. Endospores are really tricky, as many people have experienced. So only 2 samples have been reported with endospore release that I could find. And no arthrochonidia yet have their transcriptomes have not been sampled, which I think is important since that is the infectious particle. So I wanted to help fill in some of these areas and collected 7 time points to capture more stages of this complex developmental process. I also optimized endospore release, so was able to get a number of spherule samples that were releasing endospores to help fill in that gap. And then for the first time, included arthrochonidia as well, so we can start to get some insights into the infectious form. And since my interest is really in comparing the morphologies and the transcripts between them, I wanted to use the same media for spherules and hyphae to at least eliminate some of those variables. And I'll just say that my wild type is the Posidaceae silvera strain for this whole talk. So the first thing I did was I needed to optimize spherulation conditions to allow for precise comparisons. So I slightly modified converse media and was able to, from arthrochonidia, see spherules grow, release endospores with minimal hyphae in these cultures. And then by just changing the temperature from 39 to 25 and CO2 from 10% to 0, the wild type then forms hyphae instead in the same media. Additionally, to try and tease out what is related to hyphae and spherules and not related to the temperature and CO2 differences, I also leveraged a RIP1 deletion mutant, which has been published before. It lacks the critical transcription factor RIP1 that's required for spherulation in Coccidioides. So in the same spherulation conditions, it will grow as hyphae and similarly grows as hyphae in the wild type hyphal growth conditions. So I'm going to start with this heat map here of a spherulation time course from arthrochonidia through day 6, just with wild type. And here, each line is a transcript. Yellow is highly abundant, blue is very low abundance, and hopefully you can appreciate there are vast changes from arthrochonidia through the spherulation cycle with multiple kind of phases of different transcriptomes. So this is a really large-scale remodeling, which fits with a pretty complex developmental process. I wanted to narrow down to some key lists of transcripts. And so I leveraged the fact that I had multiple time points and wanted to tease out transcripts that had consistent behavior across all spherules or all hyphae. So here in the purple, I defined morphology-dependent transcripts, meaning there were 551 transcripts that were consistently different between wild-type spherules and wild-type hyphae. So some of the hallmarks of serialization. And then given how important RIP1 is to spherular development, I also wanted to define RIP1-dependent transcripts. And so I found 452 of those that were dependent on RIP1 in spherulation conditions, and 262 that were dependent on RIP1 in hyphae. And in particular, draw your attention to this 152 at the overlap. These are morphology-dependent transcripts that are also RIP1 dependent in spherules. And I think that in that shorter list, we have a lot of good candidate virulence factors to study. Then to dive a little bit deeper into the RIP1 data set, I divided it out by time point. And so here in purple are genes, transcripts that are induced by RIP1, green transcripts that are repressed by RIP1. And the first thing that jumped out was a remarkable 3,500 or so RIP1 dependent transcripts in Arthrochonidia, which we hadn't looked at before. And I think kind of surprised us that RIP1 seems to be playing a really important role in this form of the fungus. Over spherulation, I saw an increase in RIP1 induced transcripts through most of the time course until a day after endospore release started, kind of a constant RIP1 repressed number. And different in hyphae, we saw a decreasing number of RIP1 transcripts over time, implying that the wild type and the mutant transcriptome really converge in these hyphal growth conditions. So given the number of genes that are dependent on RIP1 by RNA-seq, I wanted to dive deeper and see which of those are directly bound by this transcription factor and are direct regulatory targets, because those are probably the ones we should study next. And so I implemented ChIP-seq for RIP1, and this is an example of what the data looks like, actually looking at the RIP1 gene itself here. So the promoter is on this side, and the 3' region is on this side. And in yellow here on day 1, and then more so day 2 and 4, hopefully you can appreciate robust binding by RIP1 at its own promoter, and to some extent also in the 3' regulatory region with really minimal signal from hyphae. So I took this ChIP-seq data and I went back to those shorter lists that I'd made from the RNA-seq and asked how many of these 452 spherular RIP1 dependent genes are direct binding targets of RIP1. And we get a little over 60% are directly bound in the spherular group. And to contrast that, in the hyphae RIP1 dependent, it's a much fewer are direct binding targets. So I think RIP1 is playing a very important and direct role in spherules and a more indirect role in hyphae, even though it still changes the expression of a number of genes. And then finally, I wanted to systematically look at each gene whose promoter was bound by RIP1 at some point in our data sets. And I grouped them in this upset plot here, which I'll take you through. So down here are the data sets of ChIP-seq, all of the different time points of spherules and hyphae where we detected RIP1 binding and their relative sizes. And then the genes are grouped across the top bar into different categories, and a gene can only fall into one of those categories. So what we noticed is that there are genes whose promoters are bound by RIP1 only in spherular time points, highlighted in red. And then there are a second group of genes whose promoters are bound in both spherular and hyphal time points. And it's kind of a strange, it brings up the question, how can one transcription factor regulate two pretty different subsets of genes in different morphologies? And I think we have a clue from the motif analysis, where when I looked at the promoters of these genes that are bound by RIP1 in hyphae and spherules, I found this promoter, which is the canonical RIP1 motif as discovered in a number of other fungi. When I looked at the spherule only bound group, a totally different motif was present in their promoters that bears no resemblance to the canonical RIP1 motif. So I think in this serial hyphal-bound group of genes, RIP1 itself is recognizing the DNA, whereas in this spheral group, a second transcription factor is likely involved that recognizes this motif and recruits RIP1 to that promoter itself. So I think we're looking at a complex regulatory network here. So this is the model that we proposed in the paper, synthesizing a lot of data that I don't have time to talk about, but as I was telling you, we think RIP1 controls one regulon that's morphology independent in both morphologies, and with a transcription factor, also induces serialization, and then has a great effect on arthrochonidia, although we hypothesize it's through an additional regulator. In a collaboration with Mark Voorhees, a bioinformatician in the SIL lab, we annotated all the putative transcription factors in Coccidioides and found, interestingly, that the majority of transcription factors are expressed in spheral and less are expressed in hyphae. So I think implying an even more complex regulatory network in the spheral morphology. And then to leverage the number of samples we had with endospores, we proposed a short 20 transcript list of endospore-associated transcripts that were consistently found in these endospore-containing samples. And I'll point out one of them is MEP1 here, which Dr. Hung has published about and has a known role in endospore biology, which was a nice validation to see on the list. And additionally, there's 2 other secreted proteases here as well, which really piqued my interest. And then finally, we wanted to propose a list of putative secreted effectors, and we defined these as proteins that are predicted, that have a signal P, so they're predicted to be secreted. They have 4 or more cysteines, which is a hallmark of plant fungal effectors, and they have consistent spheral more than hyphal expression. And I think quite astonishingly, 6 of the 14 that we found are also secreted proteases. So I think that's an important area of study in Coccidioides biology. And then just in the last minute here, we wanted to validate this resource as a tool for discovering novel biology. So I picked a cluster of 6 genes that jumped out from this data because they had this interesting expression pattern of high abundance in arthrochonidia, dropped during early spherulation, and then returned to high abundance once endospores were present in the sample. And the 3 across the top here, DTR1 and DIT1 and 2, their orthologs are involved in the synthesis of a dityrosine layer in Saccharomyces in the ascospore. So it fit also with a role of arthrochonidia. So I deleted this cluster all at once since they're next to each other and used those mutants to do transmission electron microscopy of the arthrochonidia, and compared to wild type, which had more distinct layers and the thicker cell wall, the mutants had thinner cell walls and less numbers of layers on average. And here's a quantification of the cell wall width. So we're excited, hope this resource will be useful to everyone in the field and can motivate some of these hypotheses in testing. So future directions, we definitely need more transcriptomic studies in other species. This is just one strain, and I know the UCSD group is working on this, that we're excited to track down that additional spheral regulator by doing DNA motif pull downs to help identify it, and then to dig more into the role of this 6-gene cluster in virulence and interactions with macrophages. And with that, I want to acknowledge my lab and collaborators, and I'm happy to take questions.
Okay, questions? I think I'll just start with a comment. I think this work is really important because it reveals potential targets, additional targets for drugs that we can use, not only against cocci, but other fungal pathogens.
Yeah, great talk. That's cool. I was curious about so you changed the temperature and the CO2 to get them to spherulate, you said. Mm-hmm. Then is it up or down?
Yeah, for spherulation, I should have put it as text. So we do 39 degrees centigrade and 10% CO2 is how we are able to induce serialization in the lab, it's higher than you would expect, you know, in a human lung outside of a dramatic fever. So we're probably, my hypothesis is we're missing some cues and we kind of have to give extra signal to get them to grow as cereals in the lab.
But then in the field, like spherulation, we think that happens in the soil, right? Or
we think it's only in the host.
It happens in the host.
Exactly.
Okay.
So probably closer to 37 body temperature is what we would expect.
But we're thinking that if there's not a human, if the natural reservoir is not humans, right, some other mammal.
I think generally their body temperatures are similar. Like the mammalian body temperature is generally 37. So it's probably that temperature is one cue of, I would guess, many that you're inside of the mammalian host.
But yeah, so maybe it gets at like trying to look at which hosts would run a little hotter. I know like cats, for example, run hotter than other, I don't know.
Interesting. I can't say that I have thought much about that,
Okay.
but that's an interesting point.
Birds are really hot.
Oh, cool.
Run like 108, and we have yet to discover it in birds, to the best of my knowledge.
Yeah.
And I think there are certain models, like you'll see a talk tomorrow about co-culture with immune cells where 37 degrees is sufficient. So I think it just depends on exactly what cues you have in the media and surrounding cells.
Cool. And then, yeah, just quickly to the proteases you were saying, like what, I mean, are those specific to certain types of proteins? How does that work?
Yeah, so proteases, I mean, many pathogens have secreted proteases and they definitely have ample precedent to be involved in virulence. It's something that's been of interest, I think, in the cocci field for quite some time because there are two protease families that are genetically expanded in cocci compared to a lot of other fungal pathogens. And so it's something, I think this adds additional evidence that we need to be looking at those families, and something I'm actively studying and will refrain from talking your ear off about it right now.
So when you were making the spherules, were they, I didn't quite understand, did you do that only from the arthrochinidia?
Yeah, so I have, the way I start these serialization time courses is from arthrochinidia. I think it's possible to grow hyphal cultures long enough that they will make arthrochinidia kind of every other segment, and then those can transition into spherules, but I don't believe, at least I haven't observed, and correct me if other people have, that the hyphae itself can directly transition into spherules. They have to kind of go through the spore phase first, I believe.
Hyphae can directly transition to spherules. It's awesome. Come see my poster, the one that looks like a crime scene. Because I was reading up on early literature to figure out how can we grow it easier in spherular form. And when you read the 50s and 60s, I'm not sure why we ended up on converse, because there's a lot of other things that were used. And I tried some things because when I saw one of those old micrographs of spherular, it blew my mind because, of course, if it's in a happy place, why wouldn't it go straight from hyphae to spherule? It's fascinating. And obviously going from arthrochinidia, that's what we breathe in. But I think there's other biology there that might give us clues into where it is in the environment, because does it want to be a spherule? If it has all these transcription factors, maybe that's what's its fanciest thing. I struggle with this, even though I'm a laboratorian, clinical laboratorian, so I'm more in diagnostics, but I think the rest of this has a lot to say about how we can deal with this disease. So really cool work. Thank you.
I'll have to check out your poster. Cool.
So yeah, Christine, really nice work. So I think what everyone's dancing around is that coccidioides is not thermally dimorphic. It's much more complex. So has this been compared, say, to histoplasma, your heat maps and so on? How do those relate?
Yes, we've definitely compared. There's a number of shared orthologs, and then, as you might expect, a number of unique transcripts to coccidioides that change with serialization, given it's a unique morphology. I think the regulation is also very different because I was showing you like RIP1 affects hyphae in addition to cereals, and this is something that's already been published too. You know, that is really different than histoplasma. And histoplasma, RIP1 controls the entire yeast transcriptome. And so the fact that we have this key regulator that doesn't explain all of the serialization is interesting. It seems like it's more multi-layered in cocci than other thermal dimorphs.
Okay, we have one question online. Is the DIT or DIT cluster deletion attenuated in mice?
We have not tested that yet.
Okay.
Yeah.
Okay, great. Maybe just one last quick question, Amy.
Quick. When you did, when you did your arthrochonidia, I mean, if you infect mice, for example, with your arthrochonidia, it's about 96 hours to get to mature spheral and rupture. So at what point is your arthrochonidia being tested? Right? In that 96-hour window, or I don't know. So that's question one that should be very fast. And then the other is, did you see upregulation when you got into hyphae? Is where I'd expect to see upregulation of the enzymes that will digest keratin, for example, right? If that's happening as the mammal dies. So
yeah, so I think I'll start with the first one. So the arthrochonidia samples are all taken at the beginning of the time course. It's what I use to start cereal growth. So essentially there's not any time component to them. They're like a time 0. And then the keratinases, so one of the expanded protease families I mentioned, the S8 or serine protease subtilase family is the one that's like hypothesized to have keratinase activity. And that the expression pattern within that family, there's some that are spheral predominant, some that are hyphal predominant. So I think likely it's expanded and has, like, my hypothesis is it gained multiple functions, but one of them is likely keratinases. Yeah. Thank you.
Thank you. Okay, so this is our last